gene expression. Our next example is non-gene expression. This one is regulation of enzyme activity. So again, you have your G protein coupled receptor, you have your ligand binding to that. When the receptor is activated, it's going to activate the G protein, which then swaps a GGP to a GTP. The alpha subunit of the GTP activates the enzyme medullary cyclase, which then will take ATP and make cyclic AMP out of that. None of that is different than the previous example. Cyclic AMP then activates an enzyme, which then activates other enzymes, etc., etc. So what you see here is that you have an inactive protein kinase A, which with cyclic AMP becomes activated. So now you have an active kinase. And what do kinases do? They phosphorylate other proteins. So this kinase is going to phosphorylate an active glycogen synthase and deactivate that one, and phosphorylate an inactive phosphorylase kinase and activate that one. So why is it important that you deactivate this enzyme? That's the enzyme that is going to use glucose and make glycogen. And the whole point of this cascade is to actually release glycogen. And so you don't want to use up release glucose, so you don't want to use up the glucose to make glycogen. So you inactivate the enzyme that uses glucose, and you activate the cascade that is going to release glucose. So the active phosphorylase kinase is going to activate another enzyme, which is glycogen phosphorylase, which is going to break down glycogen. So you deactivate the enzyme that makes glycogen, and you activate the enzyme that is going to break down glycogen. And the final response is releasing glucose into the bloodstream. Now, the reason that I wanted to show you this example is because it's not involving gene expression, it's just regulation of enzyme activity, and also because it's a phosphorylation cascade. This protein phosphorylates something else, this can phosphorylate something else, and at each of these levels, you could amplify the signal. So if you look at one molecule of the hormone, the ligand, when you start, you end up with 10,000 molecules of glucose being released into the blood. So at several of these steps, you have amplification. And you could also have multiple regulation points. So this pathway could potentially interact with other pathways that could provide inhibition or activation of other enzymes. So we are looking at it at this pathway as if it was simple and linear, which it can be, but it could be branched and interact with multiple other pathways as well. So both multiple points of regulation as well as amplification, and you can see the numbers on the right side here. So far, so good? Great. So the ultimate response, some regulation of some kind of cellular activity, and that could be through gene expression or could be through regulation of enzyme activity. And we've seen both examples. So we're going to take a look at other secondary messengers. So calcium could be another second messenger, and calcium is regulated through the activity of multiple calcium pumps. So if you look at the cytoplasm of the cell, typically has very low concentrations of calcium because there are pumps in the plasma membrane, in the mitochondria, and in the ER that pump calcium out of the cytoplasm into those compartments. So the cytoplasm is maintaining very low concentration of calcium. So you have these reserves of calcium outside of the cell, inside the mitochondria, and inside the ER that could be used if there's a signal that will cause the release of calcium. So you have to think, why would you want to maintain the calcium concentration here really, really low?
Electrochemical gradient.
One is the electrochemical gradient, because that way, if a channel opens, calcium can rush back into the cytoplasm. Any other reason? Yes, sir. So the negative charge inside the cytoplasm goes into the electrochemical gradient that somebody else mentioned. So if you have an electrochemical gradient that favors the movement of calcium in, if a channel opens, then calcium will flow into the cytoplasm. There are a couple other reasons, and we're going to come back to that. So let's take a look at examples of signal transduction pathways that involve changing the concentration of calcium. So let me show you one example. I'm going to show you this root rabbit tropism video. So this is from Indiana University, and one of my former students then was a grad student here and made all kinds of movies like this. These are old time lapse videos. And this is a corn kernel that started to germinate. So here's the corn root. And what they have done is it was germinating downwards, and then they turned the corn. So now the root is sideways. And they're filming to see what happens to the root over time. And this has to be done in the dark, so they had to actually use special cameras so that they could videotape in the dark. And you can see that the tip begins to bend down. The older part of the root doesn't bend, it's only the tip that grows down. This is actually a growth movement. So the mature part of the root doesn't bend. Only the new growth is moving downwards. And the question is, well, how does the root actually sense this? Sense the direction of gravity to know that it should grow this way, as opposed to just keep growing the way it was growing before, which was that way. So you've already seen the organelle that's involved in that. I'm going to remind you of the lab that you did with the microscopy lab. So when you look at a root, here's the root tip. The root tip has what we call a cap. Now the cells in the cap have some organelles that you have heard of before. Amyloplasts. You saw them in the lab in the tubers of potato. Tubers of potato are actually stem, they're not root. But the root cap cells have them also. So what are amyloplasts? Yes, ma'am. They store large granules of starch and you stain them in iodine. And so the thing about starch granules is that they make amyloplasts very heavy. And so if you were to look at this cell, so I'm going to take one of these cells and make it much bigger. Inside the cell wall nucleotide, so here's the cell wall, here's the plasma membrane, and then you have these amyloplasts. These amyloplasts are heavy with starch and they fall to the bottom of the cell. And the pressure of the cytoplasm being exerted on the lower membrane causes some pressure-sensitive channels to open. And it turns out that calcium is involved in telling the roots which direction down is. And there's also a hormone called auxin, which you don't need to know right now, but you will in Bio 102. Auxin is also involved in this process. So as the root is growing down, these are falling down, they send the signal down is this way, and the root keeps growing. But what happens if you turn this sideways?